Hi everyone, my name is Mary Park. It is week 4. This is lab 6, biochemical testing for microbial identification using methyl red and Voges Proscure and catalyst testing. Alrighty, so with this test, we'll be using active cultures such as our E. coli and our S. epidermidis. I'm going to go ahead, okay, so like I said, the results have been done, so which means that I'm going to show you exactly what's needed to do the experiment. So with the tubes, you want to make sure you label it to, let me just turn the video around. You want to make sure that it is MRVP and RVP, and then you want to make sure that you label it to E. coli and S. epidermidis. And we're going to put 4 drops of E. coli and 4 drops of S. epidermidis into the MRVP tube, and we want to make sure that we incubate it for 48 hours, and my incubation is the pantry in a box. Then I'm going to go ahead after 48 hours, I'm going to check for any bacterial growth, which should appear as turbid. If no growth is observed, then continue to incubate for another 24 hours. So then I'm going to go ahead and carefully pour half of the incubated MRVP broth, and I'm going to label the test tubes. Obviously, one's obviously E. coli and then one is S. epidermidis. And then I'm going to add 7 drops of your methyl red reagent into the E. coli and S. epidermidis. And then after that, you should immediately see the color of the broth. After adding the drop, you should be able to see if there's a color change. So if the test is positive, the broth will appear red or pink on top of the actual test tube, which I will show you the results in the next video. Okay. With the Voges Proscure test, I'll be using reagents such as Barrett's A reagent and Barrett's B reagent. I will be pipetting at least 12 drops to each tube, and then I'm going to let it, for B, I'm going to make sure that I will expose it to oxygen for 30 seconds and then place each tube in a holder for 30 minutes. And then after that 30 minutes, I should be able to observe the test for any other color change. A color change obviously will signify that there is a positive test. So for the motility test, with the motility test, I'll be dealing with, let me show you the tube. With that, I'll be dealing with the S. epidermidis and the E. coli, which I will be collecting the colonies from this agar plate. Okay. And I will be using a paper clip to stab the actual into this agar motility media, and then I'm going to incubate it for 48 hours, and then I will show you the result in the next video, but that's it. Thank you.